session right now, which actually involves what we are more or less expecting, the pipeline update on Friedreich's ataxia and where we are heading. I mean, the science is great. We are very fortunate to have all these scientists on board with us, but we need also to understand the pipeline and where this it stands at the moment. Παρακαλώ, περάστε μέσα να κλείσουμε και να ξεκινήσουμε τις εργασίες μας. Okay. We are... Okay, and without further ado, let me introduce Myriam Rai. Dr. Rai, she is the Director of Global Relations and Initiatives at the Friedreich's Ataxia Research Alliance, which is the US patient advocate organization leading globally the world effort for fighting FA and finding an effective treatment and ultimately a cure. Myriam, the floor is yours, and you will introduce to the rest of the session. Thank you.
So, good afternoon, everyone. Well, I will reiterate what all the others have done, thanking the association, Vasilis, but also all the other members of his team who are not on this place of the stage today, but who did a very great job. And I can tell you how complicated it is nowadays to organize a meeting, partially in person. It has been really challenging. And don't forget the challenge aligning all the different time zones of the intervenants. So thanks to the association. And rather than introducing the pipeline, which Jen Farmer, my colleague, will do, I would like to start the pipeline on FA clinical research. Okay, shall we wait? No.
We can start with the introductions and
okay. Because my message is mostly meant to you here as FA community. And yeah, I will wait. This is why. Yeah, no problem. Yeah, thank you. So I would like to say thank you to the FA community. Thank you for all of you present here, FAers, parents, caregivers, researchers. Thank you to the FA community for your support to FA research. You are supporting the research whether by helping fundraising, and we heard this morning how HEFA has supported the genetic lab at Agent Hospital to keep the adequate equipment needed for the diagnosis of Friedreich's ataxia, whether by raising awareness, and don't forget May is the month of Friedreich ataxia awareness, so thank you for raising awareness like this event. Thank you mainly for participating to research studies. And I'm saying clinical studies because I know, unfortunately, not many clinical trials are happening for FA in these regions. And I can tell you there is a global effort to change the situation. And we are looking forward, to seeing clinical trials happening here. But thank you for participating to the clinical studies. And I would like to insist on how important is the natural history studies on FA, like the EFACT Center here in Athens. This is providing a structure and a very valuable registry to better understand Friedreich ataxia, to build and be ready for better clinical trials. Thank you also for those who are registered at the Friedreich ataxia patient global registry. Soon it will be available in many languages, including Greek, and data collecting from there help also to build better clinical trials. So again, thank you for being an active member to the FA research. We have achieved together, and thanks to your efforts, some milestones and some important milestones to the journey on the development of treatment for FA. The timing is now very exciting. We have a lot of things getting ready, being developed. We will have today an outline, an overview of the FA pipeline that will be introduced by FARA. And then we will hear another perspective, an industry partner perspective of a specific therapeutic approach. And at the end, we will have the clinician perspective on treatments under development, but also how all these will be integrated into clinical practice. So without having extra delay, I would like to introduce my colleague, Jen Farmer. Jen is the Chief Executive Officer of FARA, that's the Friedreich Ataxia Research Alliance in the US. Jen has many, many long years in FA. She's very close to the FA community. Jen has a major as a genetic consultant, so she knows very well the disease. And I've been into FA field since 21 years, and Jen was already there. So you can really trust what she will be telling you today. Jen, the floor is yours. Thank you.
Thank you so much, Myriam, for the introduction, and would just like to join you in thanking all of the organizers of this conference for putting together, a really spectacular agenda. I've been able to listen in for the past few hours to a lot of the research talks that you've heard, and really appreciate being able to join the conference today. So thank you all so much. Let's see, hopefully you can see my slides. And let me just, there we go. So before I get started on the treatment pipeline, I thought it might be helpful to some of you for me to tell you a little bit more about the Friedreich's Ataxia Research Alliance and what we do, because we really hope that we'll be able to work more closely with AHEFA and your community, both on the research as well as, basic research, as well as the clinical research that Myriam was talking about, the natural history studies and bringing more clinical trials to Greece in the future. And so the Friedreich's Ataxia Research Alliance was founded in 1998 by patient families affected by FA, as well as researchers. And the focus of the Alliance is really to advance the research to find treatments and a cure. We believe that the way to do that is by raising funds and bringing people together who can both do the research as well as support the research. And so a lot of what our organization does is very research focused. And we try and do this in several different ways. And these are the strategic pillars that you see here. So we assemble conferences like this one to bring people together to share information, to make sure that there's an opportunity for, education as well as knowledge sharing. We don't want research to be in a silo, and we know that it's important for researchers to also engage with the patient community and the clinical community. And that's what these kinds of conferences are so important for doing. We also, spend a lot of time with all of our partners trying to build collaborations. We know that, I think when you saw Dr. Dettori earlier today talk about her research, it was really impressive, the number of researchers both in Australia and outside of Australia that she's collaborating with to do her research. And those kinds of collaborations we believe are really important. We also know it's important that we build collaborations with regulatory agencies like the European Medicine Association, and collaborations between our organizations. And that's one of the important roles that Myriam is fulfilling for FARA throughout Europe. And so, we really believe in partnership and collaboration. The other aspects of what FARA does is we directly fund FA research, and we fund FA research around the world. We do that through research grants to identify specific, to answer specific questions, to understand FA better, whether that's the cells in the dish or studies in people like natural history studies. We also believe that it's important to create resources for the community so that we can speed up the research process. I think one thing that we hear from patient families all the time is that the research goes so slowly. And so one thing that's really important to our organization is identifying ways to make the research go faster. And one way to do that is to create resources that are available to the whole research community so that we reduce the time it takes to do that research. So a lot of the cell models or cell lines that are needed to do research, we've put in place repositories where cells can be shared amongst researchers. Animal models are in places where all researchers can access them. We've also put in place resources like the patient registry that helps everyone kind of understand where individuals with FA are in the world and allows us to recruit clinical trials more quickly. So these patient registries are incredibly important because they identify where people with FA are globally, but then allow us to communicate directly with the community as well as recruit for clinical trials. And as Myriam was saying, the natural history studies are incredibly important. We're not able to do clinical trials if we don't understand the disease in terms of the clinical symptoms, how they progress, and have ways to quantify or measure that progression over time. We need tools called outcome measures and biomarkers that can be used in clinical trials to measure how the disease changes over time. And your participation in these kinds of studies is critical for us to be able to do those trials. And so FARA has invested a lot over the last 20 years in natural history studies, biomarker studies, to help facilitate getting clinical trials going and improving our ability to do those trials reliably and efficiently. So that's just a little bit about FARA and what we do. I'll switch gears now and just talk about the treatment pipeline. Before I jump right into the treatment pipeline, I just want to say a few things about how we think about developing therapies for FA. When we think about developing therapies for FA, there are multiple different ways to approach treatment, and we believe that this is incredibly important. We can target the frataxin gene. We can think about targeting the frataxin protein, the mitochondria where frataxin serves its function. And we can also think about symptoms that affect individuals with FA, and can we treat the individual symptoms that are occurring? So when we try and think about identifying these treatments, I like to think of this as sort of a cascade, but a lot of the research that we've been doing for the past 20 years is to identify not only treatment approaches, but what we call targets. So very specific genetic or molecular targets that we can make drugs from that will ultimately reverse some abnormal biology or some, an abnormal cell process and ultimately lead to improvement in symptoms. And so the kinds of questions that we ask will inform identifying these targets. And so if we start up here with the FA gene and the mutation that's occurring in FA, which is the triplet repeat expansion, we ask questions like, how does the expansion occur? And how does the length of that GAA repeat expansion impact the phenotype or the symptoms and the progression of the disease? And so are there treatment approaches that can target this GAA expansion itself, and will that be meaningful to treating the disease? We know that the GAA repeat expansion silences the transcription of the gene and reduces the amount of frataxin protein that gets made. So a lot of research has gone into trying to understand how the repeat expansion silences the gene, and are there genetic or small molecule targets that we can identify that will reverse this silencing. Then, kind of going to the next level down, if we think about the frataxin, the function of frataxin in the cell, is that something that can be substituted by another protein or another drug? Frataxin functions in the mitochondria. And so a lot of work has also gone into understanding what frataxin does in the mitochondria and the consequences of frataxin being lost. And so, the oxidative stress or mitochondrial dysfunction, are those things that can be intervened upon? Can we borrow therapies, that are being developed even in other diseases that can reduce oxidative stress or improve mitochondrial function? Then trying to understand how all of these consequences, play out in the cell or in the organ tissues. And so, how do these abnormal processes lead to cell death? Why are some types of cells more vulnerable than others? And then how these changes play out over time in terms of, changes that might happen very early in development, as well as the progressive changes that take place over time. And then finally, really trying to understand how this all translates to the symptoms that people have with FA and which of those symptoms are most important for developing therapies. And so this just should hopefully help you understand how we think about trying to develop treatments for FA and sort of the basis for this treatment pipeline. And, with all the work that's been done over the last 20 years, the good news is that we've learned a lot, and there are now lots of different ways in which we've identified where we might be able to intervene in the abnormal, processes that are going on from the mutation all the way down to, the tissue dysfunction or the organ tissue impact in the disease. So, for example, when we think about the repeat expansion, there's work now going on to try and cut out the repeat expansion or edit the mutation. We understand a little bit more about the silencing of the gene and specific drugs. There's a class of them now entering clinical trials called synthetic transcription elongation factors, or SYNTEFs. And these SYNTEFs directly interact with the FA gene and reverse the silencing to allow more frataxin protein to be made. There are also now therapies entering the clinic that are both trying to replace the abnormal FA gene as well as the abnormal protein. There are many drugs now that have been identified that can improve mitochondrial function and reduce oxidative stress, and you'll hear a little bit more about those today, as well as looking at trying to protect the cell from those consequences. And symptomatic therapies that you also heard about today, with things like trying to regulate iron or even exercise as a therapy to improve symptoms of FA. And so when we take all of this and try and put it into one picture, and try and put it into a picture that tells you where in the process are different treatments for FA, we this is what we call the treatment pipeline. And so across the top is sort of the stage of development. And starting on the left, this is when things are very early. So in what we call discovery phase, and so that's being tested in the lab, in cells, typically in the dish, or sometimes in animal models. When particular treatment approaches kind of move from the discovery phase and we say, yes, we think this is a potential treatment, it moves into what's called preclinical development. And preclinical development is where you do a lot of testing in terms of dose and safety of the therapy to understand how you will test the therapy in humans and people. There are regulatory steps that occur along the way that are necessary where new drugs get approved. I apologize that I'm using US language in this pipeline slide, but both in the US and in the EU, new drugs have to be reviewed before regulatory authorities, the FDA in the US or the EMA in Europe, before they can enter human clinical trials. And so these are important steps that occur in the process of developing drugs. And then human clinical studies occur, and they occur in different phases. We typically start with safety studies and then move on to what we call efficacy studies to understand how the drug is working and improving, and if the drug is improving a specific symptom in the disease. And then if we see that the drug is both safe and having a positive benefit, it again goes through a regulatory review process before it can be ultimately available to patients diagnosed with the disease, so that doctors can write a prescription and give you a medicine. And so this, here we're just kind of trying to show you where in the process different treatments are. So each of these bars is, where the arrow ends is meant to tell you where in development the treatment is. Then going down the left-hand side are those treatment approaches that we just talked about. So going from, and now we're going in reverse order, so I apologize for that, but here starting at the gene therapies through the mitochondrial-based therapies. So the mitochondrial therapies are the ones that are the furthest along in development. And the first bar here is a drug called omavaloxolone that is being developed by a company called Reata Pharmaceuticals. And this drug has gone through multiple clinical trials, and there was a positive result from the most recent trial. And so Reata has submitted to the Food and Drug Administration in the US for approval of OMAV as a treatment for FA. And so this drug is sort of the furthest along in the process. You're going to hear from Dr. Matt Klein from PTC Therapeutics about vatiquinone. Vatiquinone is also in current clinical trials, and it's at the stage where it's being tested for efficacy to see how the drug improves the neurologic symptoms in FA. There are several other drugs that are currently in clinical trials that are targeting reducing oxidative stress or improving mitochondrial function. These are all very small studies, typically about 10 to 20 people, and kind of looking for a signal to see if the drug is having the effect that they think it should have. And then if these drugs have a positive signal in these small early stage studies, they would then move forward to much larger, multinational studies to really understand if the full picture of the treatment effect. But these are, several different drugs that all focus on mitochondrial function and trying to protect the mitochondria when frataxin is deficient. You heard earlier today about a drug trial that will soon start in France that is looking at iron regulation. There are several drugs that are currently in development that are looking at pathways that are abnormally regulated because frataxin is deficient. And so one of those that recently completed a clinical trial in Europe is laraglitazone, and that drug is being developed by Minarex. They did a small phase 2 study and saw some positive signal. And so hopefully we will see laraglitazone go into a bigger multinational study, either later this year or early next year. There are several drugs that are in development right now that are focused on the frataxin protein, and one of them is called CTI-1601. And with CTI-1601, the frataxin protein itself is being delivered. And there's a special carrier molecule that is attached to the protein that allows the protein, when it's injected, it's given through what's called a subcutaneous injection. So kind of like insulin is delivered for diabetes, the drug is injected into the skin, but it then disseminates throughout the body. And the carrier molecule allows frataxin to get into the cell and into the mitochondria. And this is being developed by Larimar Therapeutics. They finished a first-in-human study last year and currently evaluating longer-term safety with the drug before they continue to go into the next clinical trial. There are several different treatment approaches that are trying to increase the frataxin gene expression. So, reverse that silencing that I talked about earlier. And the synthetic transcription elongation factor drugs that I mentioned are also called gene tags, and they are being developed by Design Therapeutics. And these drugs have just entered the first clinical trial. And so people are getting just a single dose of this drug to test its safety and to figure out what dose might work to increase frataxin. So this just started about a month ago. There's a clinical trial ongoing in Australia right now to look at a high dose of a supplement called resveratrol that they've been investigating now for several years that can increase frataxin levels in the cell and should have results of this study later in the year. And then there's a lot of activity in the gene therapy space, both replacing the gene as well as trying to edit the gene and cut out those triplet repeats. And so most of this research is still ongoing in the lab. So testing in the animal models to understand dose and safety and distribution. But you can see just by the number of companies listed here, the amount of activity that is ongoing in trying to develop gene therapy. As you heard from several of the speakers earlier today, gene therapy is challenging because we want to get the gene to many different cell types in the body. And right now, the viral vectors and the nanoparticles that are currently available don't allow for the gene to be delivered to every single cell in the body. And so a lot of these different programs have had to pick and choose kind of different approaches, both what cells they're going to target or organ tissues they're going to target, as well as how these therapies are going to be delivered. One of the programs being advanced by Lexeo Therapeutics announced earlier this year that they've gotten approval from the Food and Drug Administration in the US to begin their first clinical trial. And Lexeo's gene therapy approach is to replace the frataxin gene using a viral vector called AAVrh10. And they're specifically focused on trying to deliver the gene to the heart in FA to protect from the cardiac disease or heart failure. And they will, they anticipate starting this first clinical trial later this year. So that kind of gives you an overview of the different treatment approaches that are currently in clinical trials or soon to come to clinical trials. FARA keeps this treatment pipeline updated on our website. And so you can always visit the website. For each of these things, you can kind of click on any one of the bars, and there's a paragraph of information that tells you some background about the drug and where the drug is in its current stage of development. And just to end, you know, to tie together a lot of what you've heard today, you know, we all know that there is a high unmet need in the FA community. There are no approved therapies today. And so any treatment approach, whether it is to treat symptoms or to, you know, change the disease progression, are going to be meaningful. We believe that for FA, we're going to need multiple therapeutics to get to a treatment and a cure. And the exciting thing is that, you know, we currently have a lot of therapies in development in clinical trials. And there's a lot to be hopeful about for the future. And one of those, you know, therapies is currently under review. So we could have a first approved therapy, you know, by the end of the year. There's a lot of interest from the pharmaceutical industry. And I think this is really important for you all to know and to understand that we're not doing this alone. There are a lot of partners in this, a lot of people helping to develop therapies for FA. And, you know, what's critical to making this all go is our global engagement and partnership, and that partnership across all of our stakeholders. And so I'll stop there and thank you for the opportunity to participate today, and happy to take questions, I think, later in the session.
Thank you very much. And just let's see, the slides come up. While the slides come up, let me begin by giving a thank you to Vasilius and the honor of being able to come and present at the first Hellenic Friedreich Ataxia Society meeting. I've been involved in developing therapies for Friedreich ataxia for over a decade. And it has been clear to me since the first day I started that getting drugs for patients with Friedreich ataxia is a team effort that requires a close collaboration between scientists, patients, families, foundations, patient associations, companies, regulatory authorities. As Jen pointed out in her previous talk, there are so many people that have to work together to get this forward, and I am grateful to have been a part of this community in our efforts. Looks like I'm still waiting for slides. I will start talking and the slides will come. So as I am currently with PTC Therapeutics, we are a company based in, something's happened. ancora.
No, it's okay. It's okay. Yes, sir. Extend.
Okay, start again. Thank you again for having me here. It is a pleasure to be here. Let me begin by talking now about PTC. We are a global biotechnology company that's focused on developing therapies for patients with rare diseases. We were founded in 1998 with a very simple mission: use innovative science to develop drugs for patients who need therapies. That was our goal when we started in 1998, and that continues to be our goal today, even with our growth and success over the past 20 years. While we are headquartered in the United States, we have offices in 20 countries around the world and have activities in over 50 countries and have over 1,200 employees. But as we have continued to grow, we have continued to focus on this mission of developing therapies for patients that need it. Today, we have a very broad portfolio targeting a number of different rare diseases. We have approved therapies for diseases including Duchenne muscular dystrophy and spinal muscular atrophy, which we heard a little bit about earlier today, and have programs in a number of different diseases using a number of different approaches. Today, I'll be talking about our programs in Friedreich ataxia, and I'm very proud to say that we have two different programs, one using something taken by mouth, what we call a small molecule, and the other a gene therapy approach, which we have heard a lot about today. I'll focus first on our small molecule program, which is with a drug called vatiquinone, which is currently in development for Friedreich ataxia, as well as other mitochondrial disease disorders. Vatiquinone acts at a target in the cell that is called 15-lipoxygenase, or easier to remember, 15-LO. By targeting 15-LO, our drug vatiquinone seeks to regulate the inflammation and cell stress, which is known to be very important in Friedreich ataxia and a number of other diseases. It targets a specific pathway that's called ferroptosis, which is a big word to basically explain inflammation, cell injury, and cell death, which leads to the symptoms of Friedreich ataxia. It's given by mouth, either as a liquid or as a pill. And as I mentioned, it's right now in clinical trials for Friedreich ataxia, as well as seizures associated with mitochondrial disease. To date, over 500 patients around the world have been treated. Children as young as 1 month of age, adults as up to 68 years old have been treated with the drug, and we have learned a great deal about how the drug works. The pathway that we're targeting with vatiquinone, this 15-LO pathway, has recently been shown to be very, very important in a number of brain diseases, including common diseases like Parkinson's disease and Alzheimer's disease, but also importantly in rare diseases, including Friedreich ataxia. The way the drug works, I'll go through now. First, let me explain what goes on in the cell in diseases like Friedreich ataxia when there's high levels of stress. When there's high levels of stress, that enzyme 15-LO gets turned on. When that enzyme gets turned on, it increases cell inflammation and cell stress and decreases cell survival and leads to cell death. What vatiquinone does is it turns 15-LO off. And by turning 15-LO off, it can decrease inflammation, it can decrease cell stress, improve cell health and cell viability. So basically, it's acting at an enzyme that's turned on by the disease, and by trying to turn the volume down or turning it off, it can decrease the inflammation and stress in the cell. So the rationale, or the reason why we're developing vatiquinone for Friedreich ataxia, has a very strong scientific foundation. It also has a clinical foundation, which I'll talk about shortly. But importantly, this pathway has been shown to be very, very important in a number of different models of Friedreich ataxia in the laboratory, and also by looking at blood samples from patients with Friedreich ataxia. As we heard earlier this morning from Professor Rotig, iron accumulation is very important in Friedreich ataxia. There's a lot of iron that leads to stress and cell injury and cell death, and there's evidence of the pathway regulated by 15-LO being elevated in patients with Friedreich ataxia. So what does that mean? It means that we know we're targeting an enzyme, 15-LO, that you can see in patients is active in the disease. There's also evidence in models in the laboratory, whether those be cells in the petri dish or different animal models of disease, that again, this pathway of inflammation and cell stress that's regulated by the 15-LO enzyme is active in Friedreich ataxia. We also know that in these same models in the lab, particularly the cell models in the lab, that when we apply the vatiquinone, the drug, we see that we turn the volume down of 15-LO, see inflammation go down, see cell stress go down, and see cells survive. So when you put all of these different pieces of evidence together, it tells us that we're working on an enzyme that is important, that could be very important in the disease, and it really supports the idea that turning this enzyme's activity down can be important to cells and potentially important in slowing the progression of Friedreich ataxia. We have also had clinical experience with vatiquinone, which supports its further development for patients with Friedreich ataxia. Patients with Friedreich ataxia have been treated in some of our earliest clinical studies, which were compassionate use studies, where there were reports of improvements in patient symptoms. And we have also done a previous clinical trial in patients with Friedreich ataxia, which also gave us encouraging results to continue to develop the drug. I'll also mention that this clinical experience also not only teaches us about the potential for a drug to be helpful in the disease, but it also teaches us how to do clinical trials in a disease. I think as we've talked a little bit about today, and Jen Farmer emphasized in her talk, drug development in Friedreich ataxia is very challenging. One of the challenges we have is we don't yet have an approved therapy. So it is hard when you don't have an approved therapy to know the path to go down to get another approved therapy. And so we have many companies and many researchers have spent a lot of time trying to understand how do we design these clinical trials that we need to take to EMA and to FDA to get drugs approved. And there's a lot we can learn by talking, but the only way to really learn is by doing and by conducting clinical studies and then making learnings and then being sure we apply those learnings to future clinical trials. So as we move forward with vatiquinone and Friedreich ataxia, we are now in our phase 3 trial that we call MOVE-FA. And this trial is building on the scientific rationale I talked about, the clinical experience we have with the drug in other studies, and also allows us to apply a lot of the lessons we have learned as well as other companies have learned in doing clinical trials in Friedreich ataxia, with the hopes that we give the trial the highest chance of success. This trial is currently ongoing. It is being conducted around the world with sites in Europe, in Australia, New Zealand, United States, and Brazil. And it's basically what's known as a placebo-controlled trial, which means patients come in the trial and they will get either the drug, vatiquinone, or placebo for a period of 72 weeks. And that sounds like a long time, and it is a long time, but again, this was one of our learnings that having a longer trial is really important in being able to show that a drug works in a disease like Friedreich ataxia, which takes many, many years to progress. Our job in a clinical trial is to take a small part of the lifetime of a person and be able to show a drug works. And you can imagine if a disease is moving slowly, that the longer you can see and test a drug, the better your chance of seeing it can slow a disease. And so that was one of our important learnings that we have in this trial. After the placebo period, all patients will receive vatiquinone for at least 24 more weeks. So that means that for patients who enroll in the trial, they will have the opportunity to receive the drug, even if they go through a period of receiving the placebo first. In this trial, we enrolled patients who are 7 years of age and greater. Actually, the majority of patients in this trial are between the ages of 7 and 18 years old. This is something that we believe is important because if you have a drug that is going to try to slow disease progression, it would make sense to try to start it as early as possible before the disease has progressed. This was something we were able to do given the experience of vatiquinone in treating children with mitochondrial disease, where we were able to demonstrate that we could give the drug to children in clinical trials. The primary endpoint of the study is changes in the disease rating scale, the modified Friedreich's ataxia rating scale, and we're also looking at activities of daily living as sort of a key secondary endpoint, or another critical thing we're going to measure in the trial to demonstrate that the drug is having an important effect on patients with Friedreich ataxia. I mentioned that this study is fully enrolled, and we expect to have the results of this trial in the springtime, or what we say the second quarter of 2023. I'll now shift gears to talk about our other program in Friedreich ataxia, which is our gene therapy program. And PTC has a platform of gene therapy programs that includes Friedreich ataxia and other diseases. We've taken an approach in our gene therapy programs to do something that we call targeted delivery. You can give a gene therapy in many different ways. Many of the therapies being studied today are given intravenously. That is, that it's given like as an injection, and the gene therapy then has to travel to many parts of the body where it might work. The approach we're taking at PTC is something we call targeted delivery to the brain. And what that means is that we administer the gene therapy directly to a part of the brain where we think it will be most effective. We believe by doing that, there's many advantages. First, by delivering the gene therapy to the exact location where you think it will have the most important effect, you're certain that it's getting there. It's not like you're administering it to another part of the body and are hoping that enough of it reaches where it really needs to go. This also allows us to give lower doses of the gene therapy, again, because we know we're getting it to directly the spot, in this case in the brain, where it needs to go. Our first program using targeted gene therapy delivery is for a rare disease known as AADC deficiency. AADC deficiency is a disease where children are born without an ability to produce normal levels of a chemical in the brain called dopamine. Dopamine in the brain is very important for motor activity, movement, things like being able to raise your head, sit up, for a child to crawl, eventually for a child to stand and to walk. And children born with AADC deficiency often don't have the ability to do these tasks. And so what we're doing with our gene therapy is giving a gene using a virus, much of what was talked about earlier today, and it's given through a very special brain surgery procedure that is done now for other neurological diseases, but in this case, we're using it to deliver the gene therapy to a specific part of the brain where dopamine is really needed. And what we've been able to demonstrate is that by doing that, delivering a gene that is functioning to part of the brain that needs it, we see increased levels of dopamine and then improvement in motor function. This program is advanced now in the regulatory process in Europe. We hope to have an approval for this gene therapy in 2022. And we are taking the learnings from this program and are now moving into our gene therapy program for Friedreich ataxia. The Friedreich ataxia gene therapy program is not yet in the clinic. We're still doing important testing before we go into the clinic. But this program also utilizes the idea of a targeted approach. In the case of Friedreich ataxia, we're delivering the gene therapy to the cerebellum, specifically the dentate nucleus of the cerebellum. That's a part of the brain we know that's really, really important in Friedreich ataxia. Now, while we understand that Friedreich ataxia is a whole brain disease, there's other parts of the brains that are affected, and we also know it's a whole body disease. Many organs in the body are affected. We are taking an approach, though, of delivering the gene to one part of the brain we know that is particularly important to a lot of the symptoms, understanding, of course, that, as Jen Farmer mentioned, Friedreich ataxia will be a disease where hopefully one day we'll have many therapies, and then it's going to take many therapies to treat the disease because it's so complicated. While we have a drug in vatiquinone that gets to all parts of the body, including the brain, our approach is to say, okay, let's take the gene therapy then and focus on one specific area of the brain. And that's what we're doing in our program. As I mentioned, we're still not yet in the clinic, but we've completed many important proof of concept animal studies showing that we can deliver the gene therapy. We can show that when we deliver the gene therapy, the new gene can make frataxin levels. It can make frataxin levels that can vary by the amount of the gene that we give. And we're right now in the process of completing the remaining studies that will allow us to get into clinical studies in patients. And as Jen Farmer mentioned as well, these are studies that are typically done in laboratory animals, and what they help us understand is the exact dose that we should be giving to patients, and it also makes sure that the gene therapy is safe to be used in patients. So we look forward to continuing to advance that program and get it into the clinic as soon as possible. I'll mention that we've been able to complete the manufacturing process for material that could be used in patients. That's also another very important step in gene therapy. And we hope that once we've completed our preclinical studies, that we can then move ahead with clinical studies. And we're already talking about trying to do those clinical studies in different sites around the world. So in summary, PT Therapeutics is proudly partnering and working in the Friedreich ataxia community. We're dedicated to delivering therapies to patients affected by Friedreich ataxia. And we're excited to be able to have two different programs, one targeting the whole body with vatiquinone, the other with a gene therapy that's targeting one particular area of the brain known to be very important to the disease. Our MOVE-FA trial with vatiquinone is fully enrolled. We're looking forward to being able to share those results from that trial in 2023. And we look forward to continuing to progress our gene therapy program forward with the hope of being in the clinic in the very near future. So with that, I'll thank you all again for your attention and the honor of being able to be here.
Thank you, Matt. That was excellent. So let's move ahead. It's a pleasure to introduce the next speaker, Professor Massimo Pandolfo. He is currently professor at the Department of Neurology and Neurosurgery at McGill University in Canada. Prior to that, Massimo has started his journey in medicine in Italy, and then he moved to the US, and then to Montreal, Canada. And then he moved to Brussels for over 20 years, where I had the pleasure to work with him on FA. And last year he moved back to Montreal to continue working on FA because he didn't want to stop before finding a treatment. So Massimo, it's for you.
Kalispera, agapiti, Alinesfili, Casinadelfi. I am really...
Good evening, dear colleagues and friends. He said in Greek, the speaker.
To be part of this symposium, and I really want to congratulate for setting up this association that is putting, supporting FA patients from Greece and making them really part of the global FA community. What I'm going to do today is, let me share my screen, which I hope you can see now, is to make a number of considerations about the development of treatments for FA, and particularly pointing out certain challenges. It's not going to be as, let's say, pessimistic talk. On the contrary, and I think my first slide really shows you how I am not pessimistic, but really I would like to point to the accent of a number of challenges that are still there and that are being progressively tackled by researchers throughout the world. And we hope that we can get really beyond these obstacles to define effective disease-modifying treatments for FA. I'm saying I'm not pessimistic, on the contrary. And just to give you an idea, to put that things in context, I'm showing the slide that shows when I started my research in FA back in the 1980s. We really didn't know much about this disease. We knew that it was probably the most common inherited ataxia, at least in Europe, in people of European descent. We knew that it was autosomal recessive, but we had no idea of the genetic mutation. We had no diagnostic test. We had clinical diagnostic criteria that then we realized later on that were missing a good part of the patients. And of course, we had some supportive therapy, but essentially we didn't know anything about the mechanism of the disease. We didn't know anything about the possible therapeutic targets. We didn't, of course, we didn't have any treatment not only available, but not even in sight. And then things have changed radically, I would say, since that time. And of course, the main advance was the discovery of the genetic mutation underlying FA, which is, as you all know, the expansion of a GAA triple repeat inside the gene coding for the protein frataxin. There are some individuals who have other types of mutation affecting the frataxin gene, but so far, all of them are what we call compound heterozygous with the GAA repeat expansion, which therefore is found in each and every FA patient in the world. What does this mutation do? And I borrowed these slides from my graduate student, now becoming a PhD, Chiara Dionisi. It causes a number of changes in the structure of DNA and in the protein that bind this DNA in the cell nucleus that lead to what we call epigenetic silence. That is, this gene is not working anymore, is not making enough of the messenger RNA, and is not making enough of the protein it encodes for, that is frataxin. So basically leads to reduced level of frataxin, transcript and protein, which are the cause of the disease. Friedreich ataxia is a disease of frataxin deficiency. This deficiency is not absolute. There is a residual level of frataxin that is made, and this is very important. And we realize, we can see that some cells in the body can do pretty well with that residual level of frataxin. Others do not. So there is what we call a specific vulnerability to this partial frataxin deficiency that defines the pathological and clinical features of the disease. And how much frataxin deficiency is important for developing the disease and its entity is important can be clearly seen by the fact that you have some individuals, as it is a young man in the video here, who is in his early 20s, is on a wheelchair, and as you can see, is severely ataxic and has very low levels of frataxin. And we have other individuals like the lady here that you can see with Myriam, who is in her 70s, and as you can see, is much less affected. And the difference is basically in the level of frataxin that these patients have in their system. Now, this leads us to the first, obvious, in a way, approach to try and treat FA. If the disease is a disease of frataxin deficiency, let's see whether we can correct this frataxin deficiency. If we can bring the levels of frataxin to those of normal individuals, or at least to those of heterozygous carriers. This is a recessive disease. The parents are heterozygous for the causative mutation. They have maybe 50-60% of the level of frataxin of their affected children, and they are fine. So let's see whether we can find ways of boosting the expression of frataxin to that level. And this slide shows you some possible approaches. Of course, gene editing, you heard about it, act directly on the GAA repeat expansion and get rid of it. It's still at an early stage of preclinical development. There are a number of challenges in order to take this to clinical fruition, but there are a number of groups that are working on it. The second possibility is the use of epigenetic modulators, that is of different types of molecules that can act on that epigenetic silencing that I mentioned before. That is, those changes in DNA structure in the proteins that bind the DNA, the frataxin gene, that make it silent. And by trying to change this kind of structure, so to add at this level, one can try and boost the expression of the frataxin gene. That is, make it express, put it in the situation of making mRNA and therefore protein as if the expansion was not there. The expansion is still there, but is made, let's say, harmless by this kind of epigenetic modulators. There have been approaches in the past to use molecules called histone deacetylase inhibitors. Unfortunately, they haven't got to to the clinical phase because of issues about biodistribution and toxicity. And but there is now a phase 1 study that just started by the company Design Therapeutics that uses an epigenetic modulator, essentially a specific molecule that would boost the transcription of the frataxin gene and therefore the expression of frataxin. Just started. We will see what's going to happen. And then, of course, there are a number of other ways of acting on frataxin deficiency. Gene replacement therapy, which is the classical gene therapy, introducing by some kind of a vector a copy of a DNA fragment that encodes for frataxin, which is an extra copy compared to those in the chromosomes, and use it to make the frataxin that is missing in the cells of FA patients. There are other molecules that can induce frataxin transcription regardless of the presence of the expansion. There is the protein replacement therapy, what you heard from Jen Farmer earlier about the current studies by the company Larimar that uses frataxin fused to another short peptide that allows it to be distributed in cells throughout the body and to get into mitochondria. This is ongoing. Other inducers of frataxin synthesis, that is molecules that boost the ability of the frataxin messenger RNA to be translated into proteins, and finally inhibitors of frataxin degradation that are also being studied by some groups. All of this clearly has the goal of restoring normal frataxin level. But let's ask a couple of questions. Where do you need to restore frataxin level, and when do you need to restore frataxin levels to have maximum efficacy? And we needed to have a critical look at what we call the neuropathology of Friedreich ataxia. I will only speak about the nervous system today. I am a neurologist, but of course there are other parts of the body that are affected, like the heart, like the beta cells in the pancreas that need to be considered. But just to focus into the nervous system, we realize that if we want to restore normal frataxin levels, we need to do it in the cells that are primarily affected by FA first and foremost, and we have to do it in a time frame that allows these cells to survive, because we cannot treat a dead cell, of course. So let's see briefly what are the main targets in the nervous system for frataxin replacement therapy. So the first target is what we call the proprioceptive neurons in the dorsal root ganglia. These are neurons that are found not inside the central nervous system, but in kind of small swellings of the nerve roots near the spinal cord. This contains cells that are neuron, that are nerve cells that send their axons, the fibers, the thin nerve fibers that come from the nerve cells, to structures essentially in muscle and in other and in tendons that feel the position and movements of body parts. And then the other part of their axon, of their nerve fiber, goes into the central nervous system and transmits this information to the brain and to the cerebellum that is necessary for a good movement, coordination, and balance. So basically, these cells are key to provide that information about body part, motion, and position that is essential for coordination. And these are the first cells that suffer and eventually disappear in Friedreich ataxia. And actually, the atrophy of their central branch, what we call, which goes in the posterior column of the spinal cord, that you can see here on the right, is what gave the name of the original essay by Professor Friedreich, which was called Überdegenerative Atrophie der spinalen Hinterstränge, which means only the degenerative atrophy of the posterior columns of the spinal cord. And this really shows, and this is found very early. And one issue with this pathology, which is basic pathology of FA, is the fact that it doesn't seem to progress very much when we see the patients. It's already there. And we have neurophysiological data that we collected with a kind of a sophisticated technique, which is called magnetoencephalography, showing that there is a defect basically in the transmission of the brain of this information on body motion and position. And this defect actually depends on how severe the frataxin deficiency is to begin with. So basically to the length of the GA repeat expansion, but doesn't really change with time, meaning that when a patient becomes asymptomatic, it's probably already established. And we actually have developed, now Dr. Dionisi has developed in our lab an in vitro model in which she differentiated cells, this kind of cells, these proprioceptive neurons, from induced pluripotent stem cells that you have already heard about today. And she noticed that these cells, of course, are frataxin deficient. The green and orange and red lines show the expression of frataxin throughout the differentiation and development of these cells, and you can see that these are lower than the levels of the other two lines that are the controls. But she also noticed that these cells actually express less of the markers of proprioceptive neurons, like parvalbumin, the one that I evidence now, and the RANX3, which is a transcription factor that is important for the development of proprioceptive neurons. This just means that there is something wrong in these cells already at the time when the nervous system develops, so well before symptoms happen. And she also noticed that there is something wrong in the way that these nerve fibers grow out of the cells, that they are more tortuous, they don't get as far, and she has done some quantitative analysis on this, and even the electric properties of these cells. So these are neurons, these are excitable cells that produce what we call like action potential, electric signals, while their ability to generate these electric signals is somewhat impaired in at least a subset of these cells, showing that there is a clear developmental component. We have to keep that in mind, because targeting this type of cells when individuals are symptomatic, it may already be too late. And so we cannot expect an important correction of the phenotype by a treatment that only targets this kind of cells. Do we need to target them? I think so, because anyway, there is some evidence that the developmental story is not the whole story, and these are pathology samples from Dr. Arnie Koppen in Albany, New York, who has studied what happens over time to these proprioceptive neurons, and they keep being lost. So they develop abnormally, but they are also lost. So they are a target. But this target has to be seen in the context of something that may a bit contribute to the disease progression, but whose defect has already been clearly established even before symptoms appear. So this is clearly not a sufficient target. What we need to target then? We need to target the cerebellum. The cerebellum, and particularly the deep structure in the cerebellum called the dentate nucleus that you can see in the upper image on the left, that kind of ribbon of gray matter, it's the main output pathway out of the cerebellum. The cerebellum is a part of the brain that does certain types of computation that are necessary for coordination and balance. And then it has to communicate the results of its computation, if you wish, to the rest of the brain. And it does it through the dentate nucleus. Now, this output pathway is essentially lost. If you look at the image below, the one marked with FA, you wouldn't even see the ribbon of gray matter in the depth of the cerebellum. It's gone. The cells are gone. Now, this is, again, an early process, but it's not a complete process when patients become symptomatic. In fact, we think that the patients become symptomatic when this process starts, when the dentate nucleus has already dysfunctional cells and probably has lost already a number of cells, and the cerebellum cannot communicate with the rest of the brain, and symptoms of ataxia become very evident at that time. That's when people become symptomatic. Now, that's a valuable target, because if we intervene early enough, we can probably stop the malfunctioning and the generation of these cells and save whatever is left and make it functional again. So this is a valuable target. This is the target of the gene therapy that has been developed by the company PTC, the speaker just before me. But you have to keep in mind that there is no time to lose. This is just a small summary of a study done by magnetic resonance imaging that shows how, with time, this structure keeps being atrophied, becomes lost. So if you intervene too late, there is basically not much to do again here. I think that you understand already from the previous example of the dorsal root ganglia, this example, that intervening early is a major issue if we want to preserve maximum functional capacity in patients with FA and probably with any kind of neurodegenerative disease. So this structure is being lost over time, and the data that we have from neuropathology show that even a few years after symptom onset is already significantly atrophic. And then there are other structures that become atrophic with time, particularly what we call the central motor pathway of the pyramidal tracts. These are basically the nerve fibers that go from the cortex of the brain, where the motor cortex is, where we decide so the part of the brain that gives that starts the voluntary movement. When we want to make a movement, it's our cortex that activates and gives the order to the neurons in the spinal cord that control the muscle. And it does it through a pathway which is called the pyramidal tract. And this pathway also degenerates, particularly the longest fibers that go to the neurons that control the legs. This causes weakness. This causes muscle spasms, so this causes a number of symptoms that are mostly present in advanced FA. And in fact, we have neurophysiological evidence that the conduction through this pyramidal tract is impaired, what we call the central motor conduction time. For instance, here from the brain to the muscles in the hand, it gets 15, 17 milliseconds from a normal average of a little more than 9 milliseconds. So this conduction is slowed, the potential is lower, muscles become weak. This happens later on. So we have the third phase, the third wave, if you wish. The DRGs, then the cerebellum, and then we have this wave of degeneration of the pyramidal tracts. If we act only on the previous waves, and even if we are fully successful and we don't deal with this third wave, we will nevertheless expect to have some pathology and maybe have Friedreich ataxia that becomes a more what we call a hereditary spastic paraplegia, a condition in which legs become weak and stiff, but without much ataxia. Is still a functional, clearly causing functional impairment, but it may be less bad than FA. So it may be just a partial treatment to treat, for instance, the cerebellum, but leave this part of the pathology progress. And this clearly happens with time, because we know that the longer the disease, the longer the central conduction time, so the more impaired is conduction in this pathway. So basically, if we can make a step back, we can say that what we want to do with frataxin restoring therapy, ideally, is to restore physiologic frataxin level in all cells whose function and survival are compromised by frataxin deficiency. But we have challenges. First of all, is the limited or unbalanced biodistribution of gene therapy vectors or other therapeutic agents. They have to reach these targets, at least in the nervous system, the target I just mentioned. And then they have to direct the frataxin expression to a correct level, not too little, because otherwise, the treatment fails to do what it's supposed to do, but also not too much. We don't want it to have to, and this expression has to be balanced. We don't want it to too much in some cells and too little in other cells. And we need it to be sustained over time without too many changes up or down, because it can become excessive or insufficient over time, at least in some cell types. So we needed to, first of all, to know what the current approaches, the developing approaches can do in terms of reaching their targets, in terms of how much frataxin they use, and how long and how this behaves over time. We know that current gene therapy vectors and other therapeutic agents have limitation in reaching all relevant therapeutic targets within and out of the CNS. And therefore, we can, and that's why we think that waiting for the magic bullet that can do all of this, it may be worth starting to target only a subset of vulnerable cells. Can we cure FA piecemeal? Maybe. But of course, we have to understand that the treatments that are now being used to upregulate frataxin are probably not able to target the whole thing. Treating cardiomyopathy may give independent benefit. There is a trial that will soon start of gene therapy directed for that. To what extent, in what patients, we need to know. What can we expect by treating just the DRGs or the dentate nucleus? As I said, if you try to treat just the DRGs, probably we're not going to get much of a benefit, because these are already compromised at the developmental stage. Treating only the dentate nucleus, if you do it fast enough, we can probably stop the progression of ataxia and maybe get some improvement in the ataxic component of the disease, but may not probably prevent the later development of weakness and muscle stiffness, what we call so turn the disease maybe in some sort of a spastic paraparesis. How much we can expect from neuroplasticity from vector diffusion? We target a region, the vector may diffuse to other brain regions and have a more widespread effect. We need preclinical studies in animal models to figure that out. And of course, there is the issue of administering a second and third treatment to target additional cells and tissues, and I'm referring particularly to gene therapy and the limitation it has from the immune point of view. Is excessive frataxin expression a problem? How does it change over time? I raised this question. We know that from animal and cell culture data, there may be a 2-3-fold higher expression than normal is not problematic, but there are data that 10-fold or more high expression is toxic, at least for some cell types. This may be related to various mechanisms. Too much frataxin can actually clog the import mechanism into mitochondria, the processing mechanism that makes mature frataxin, and this may affect other proteins as well that needed to enter mitochondria and be processed. There may be protein aggregation and accumulation. We see that it affected culture cells that overexpress frataxin, have it in the wrong part of the cells, and they may be too much and form aggregates. And of course, the iron sulfur synthesis may be impaired if there is too much of one component compared to the others. And finally, there is some evidence that if you increase too much frataxin expression, you get a feedback inhibition of endogenous frataxin that may be also involved other inosulfur synthesis factor, and this may be a mechanism actually of impaired functionality when you have too much frataxin. We have to consider the immune response that can, of course, first of all, and even before you can treat, leads to the exclusion of patients with pre-existing antibodies to the virus from which a viral vector is derived. There may be ways around this, ways to immunosuppress these patients, or better to derive vectors that do not cross-react with naturally occurring viruses, so that are non-viral based, but this is all work in progress. So for the same reason with the current vectors, they essentially one shot only to treatment, you cannot repeat. And if you have treated one part of the body and then you needed to treat the second part of the body, that may be problematic. There may be loss of efficacy over time if an immune response is mounted against the cells that have been infected by the virus. And there may be acute toxicity issues. Systemic, we know that high doses of certain viral vector like AAV can cause a systemic reaction with a systemic inflammatory reaction and dysfunction of important organs like the liver. And you can have focal toxicity related to immune response, and this may actually target the dorsal root ganglia, the same cells that we try to save in FA, and there is a whole, again, current research about this. We know already from experience in gene therapy that preventive and therapeutic immunomodulatory treatments are needed. And some groups are developing approaches to that should, in principle, allow repeated dosing and therefore facilitate also the piecemeal approach to treatment I mentioned before. A few words about other approaches. You have heard about vatiquinone, you have heard about the concept, and you have heard about many other approaches that are being now in the pipeline from Jen Farmer. Basically, these are targeting the downstream consequence of frataxin deficiency. This is related what frataxin does. Frataxin is involved in iron sulfur cluster biogenesis, and if this is impaired, you have alteration of its energy metabolism, mitochondrial respiratory chain, and also energy metabolism out of the mitochondria. You have oxidative stress, decreased energy production, etc. And of course, these may all be targets of frataxin deficiency. And I will just say that there are a number of treatments, of course, you have heard about that are targeting this downstream consequence of frataxin deficiency. These are important approaches. First of all, because they're probably going to be available before some of the frataxin restoring treatments are really developed enough. And second, because they may be complementary, being used together with those to assure that the best possible restoration of function is achieved and in the shortest possible time range. I just want to mention one final thing concerning these approaches to correct frataxin deficiency. You have already heard that inflammation may play a role. And we actually did look at changes in the proteins that are present in the cerebrospinal fluid, the fluid that bats the brain and the spinal cord. We looked at if there are proteins that are present at different abundance in FA patients versus controls, and we found a number, but very interestingly, we found both upregulated and downregulated proteins that have to do with the immune system and with inflammation, showing that inflammation is clearly a component of the neurodegenerative process of FA, and therefore that we needed to include molecules or approaches with anti-inflammatory properties if we really want to have a full spectrum of weapons against the consequences of frataxin deficiency. How do we assess therapeutic efficacy? That will require a long talk by itself. We needed to have clinical outcome measures, and we needed to have what we call biomarkers. This is a long process that is basically based on the ongoing natural history studies and complementary studies. I have not time to get into this, but I'm saying that this is absolutely essential to proper design of clinical trials and to get to fruition of these therapeutic developments in terms of clinical trials, have appropriate outcome measure, and particularly understand what is a clinically significant improvement. If we give a treatment, we want to see a change in the life of patients. We now don't want to see the change in a biological parameter or in a rating scale that then is in the eye of the observer more than in the life of the observed. So that's with this kind of consideration, I will stop here. I'm afraid I've even spoken a little too long, and I'm ready to take questions together with the other speakers. Thank you.
Thank you, Massimo. Thank you to all the panelists. I hope the participants, thanks to all the panelists, have a better understanding how the work is being done towards the complicated multiple therapeutic approaches for Friedreich ataxia. I would like to ask if we have questions from the Zoom chat. Do we have online questions before taking questions from the room, or we don't have questions? Okay, great. Yes.
So thank you very much. It's a good one. Hopefully next year we have you live with us here. I have a question. Do we have a better understanding on the developmental biology of the disease? Why does it specifically affect this type of neurons and it doesn't affect some other type of neuron or another cell type equally, since it's a gene that is ubiquitously expressed, or the protein is so why does it selectively degenerate these particular tissues? Do we have a good understanding so far? Thank you.
Should I answer? Okay, the short answer is no, we don't have a good understanding. We are trying to work on it because exactly this is a crucial part of the story. Why certain cells, let's say with 20% of frataxin, which is probably what's in an average level in FA patients, why a lot of cells are fine with that and others are not. And even cells of the same category, neurons, cells of the brain, some cells of the brain are fine with low frataxin, and some cells of the brain or the nervous system are not. We are trying to figure it out, and that's why we are working with cellular and animal models, and we try to dissect really what's wrong, and what we also need is to compare different cell types and look what are the differences. Why, for instance, cortical neurons of the brain cortex, are essentially much more resistant, with possible exceptions of those that control movement, from the motor cortex. They have long axons, but long axons is not the whole story because other cells, there is still a difference even between cells that have long axons. That may be related to specific factors that are important for the development and survival of certain cell types, and that are somehow affected either directly by an impairment in iron sulfur cluster synthesis or indirectly by mitochondrial dysfunction or some other downstream mechanism. To be honest, we don't know. We need to do this work.
Thank you. Can I ask one question to Mathew also? So Mathew, thanks also for the wonderful talk and for sharing your tremendous work that you've been doing with the company. My question is regarding that small molecule drug that you're developing, and it's targeting unsaturated fatty acid oxidation, if I understood well. Do you have any input on whether the effect of the drug might be different on neuronal cells versus heart cells? For example, heart muscle primarily relies on fatty acids for energy, and the opposite, I think, if I'm not wrong, might happen under stress on the neuronal component. So can you target, the drug specifically, or is it going everywhere and it might have different types of effects? Thank you.
Yeah, very good question. The target of the drug is an enzyme and a pathway that's present in all cells and has been shown to be a response pathway to oxidative stress in all cells, which is, rather than a specific approach, we're taking a more general approach to a pathway that could be important in every part of the body that's affected by FA.
May I ask? So I would like to ask you, what kind of vector do you use for the clinical trial? Is an AAV? And what serotype is that?
Yeah, it's an AAV. We haven't yet shared what the serotype is. We'll do that once we're getting closer to going into the clinic.
Oh, okay. And one more question, actually, to both of you. Given the complexity of the regulation, gene regulation, and the multiple tissues affected, how you foresee that, or how close could be, gene therapy to work?
Yeah, I think that's a very good question. I think we are all, I always like to tell the story when I was in medical school many years ago, when the human genome was cloned, and everyone believed now that we understand genes and diseases, we can fix every disease. But obviously it's taken us a long time, and I think there is understandably a lot of excitement for gene therapy and the potential for gene therapy for diseases including Friedreich ataxia, which results from a single gene. So the idea is, if you can replace the genes that aren't working perfectly with the gene that's working well, can we fix the disease? And I think that promise remains. However, I think, as we've talked about and your research has shown as well, getting from this idea or something that sounds biologically very simple and turning that into a therapy is a long road. I think we have a lot of great minds working on this together, which I think will help. I think there's promise. I think there's now one gene therapy in the clinic that Jen Farmer shared for the heart. I think that's a great sign. I think we will learn and continue to get better and better at this. And I think it might be that the first gene therapies aren't cures, but can actually help the disease. And I think as we move forward, we can think about, and gene therapy advances, right? So that you can do repeat dosing or give one vector to the brain, one vector to the heart, those types of things that I think we would all like to be able to achieve with gene therapy. I'm not sure we're there yet, but you can definitely see, as I would say, the stones on the path to get there.
Thank you.
Hello. So discussion about therapies and let's say clinical trials. What needs to be done so we can see more Greek patients enrolled to such cases? And I mean, not a generic question, who needs to do? So HEFA needs to do something, government, who needs to do and what, if you can answer that.
Thank you for this question. Do you have anything to add on that, Mathew? Because you have experience in global clinical trials. We have another question on the same line asking what the community could do to accelerate approvals, but also accelerate access to trials globally. Since your company was in touch with different regulatory agencies and have run clinical trials globally, can you give us an insight there?
So I will say that when you do clinical trials, there's many balances to try to include as many patients as possible, but also to make sure that you can efficiently get all the patients into the trial that you need. I will say that this meeting today is, from my point of view, a tremendous success in bringing the community in the Friedreich Ataxia community in Greece together with the global community. I think it raises awareness and showing that there is structure and the ability to participate in trials. And I think you have a wonderful leader who I think is a great advocate, Vasily, in advocating for bringing trials to Greece. I will also say that the Friedreich's Ataxia Research Alliance, I won't speak for Jen, she can speak, but I think has done a tremendous job in building a global network of trial sites to help companies like us when we try to do clinical trials to already have leaders pointed out, structure pointed out to allow for doing clinical trials, which are a lot more complicated than just doing clinical medicine. There's a lot that goes into it that's needed, and the Friedreich Ataxia Research Alliance has done a terrific job in setting up sites around the world.
If I may add a comment, I think what's really important is the existence of networks of clinical sites that work together for the natural history, work together to develop and test biomarkers, and work together in order to define how to best design a clinical trial. There is a network like this in Europe, and Greece is part of this network. It's called EFACS, the European Friedreich Ataxia Consortium for Translational Studies, initially funded by the European Commission, but now living an independent life. And there is another consortium in North America, but also extending to Australia, that is essentially supported by FARA. And these two are planning to be integrated and work together in the future. And this is essential because this will really offer the infrastructure to do trials that will not exclude anybody anywhere in the world from a clinical trial, because you have, and the companies that are doing clinical trials are going to be let's say the partner of the structure. They are going to be invited to have a connection with these networks in terms not only of implementing the trials, but also designing the trials in a way that doesn't exclude anybody, and in a way that is most effective to get to to have the results that we want, to know if something works, to know if something is safe and effective. So Greece is part of the map. Greece is involved with the EFACS consortium, and the EFACS consortium and the other consortium, which is called the CCRN, are going to be more integrated in the near future and offer really the possibility to everyone to be part of this.
Thank you, Massimo. Yeah.
I would add a more complexity to the questions that got from the audience as well about pediatric clinical trials on FA. If any one of you could add a couple of words on it, on the feasibility and complexity of it, so that will help the audience to understand why not much is happening in the pediatric population. Thank you.
Well, it's not that not much is happening with the pediatric population. It is the fact that the pediatric population has specific regulatory needs. I mean, the regulatory agencies that have to authorize the trial and they eventually have to accept and the therapeutics that come out from this trial, they license them. They have specific requirement for children, and this is perfectly justified. Children are not small adults, as is usually said. They may be particularly vulnerable to certain treatments. They need to be assessed in a different way. Some of the outcome measures that we use in adults are not really that effective. So we needed to design the trial specifically for children. And we needed to make sure that something that looks safe in principle in adults is also safe for developing individuals like kids. So there are a number of issues. On the other hand, if we think that we have a treatment that may be beneficial for children and children that can really benefit from it, and I insisted on the fact that the earlier we treat, the better it is, we can have sort of an accelerated way of having the authorization of running trials in children. But again, since the patient population in FA includes individuals of many different ages, it is probably the path of least resistance to start testing and treatment in adults, getting faster approval to do that, and then extend it to children with additional preclinical studies and that are required by agency. But children are not at all forgotten, and in the end, they will be the primary target of any type of treatment that is supposed to stop or even to prevent the appearance of certain symptoms.
Yeah, and I guess I'd just add and say that there actually are clinical trials right now ongoing in kids. The MOVE-FA study is in children. Some of the other clinical trials that are ongoing include children. So, we'll continue to kind of see a mix of clinical trials, some of them starting in adults and then moving to kids. But it depends on the treatment that's being tested and what potential therapeutic impact we think it'll have in terms of, what the risk benefits might be, to sort of figure out where, what the best population is to test the drug to start. So there are going to be different approaches for different drugs. And I think that's the other thing that's important to keep in mind.
Yes, I mean, the PTC trial is above age 7, and the MODERATOR trial started at age 12. So we were including children and at least adolescents in a number of trials that have already been.
Thank you both. This is very helpful, mostly for the parents present today. I think we have one last question from the audience, and then we should move to the last speaker of the previous session who would like to join us.
So you said there are no one is to be excluded from the trials. Once they are also here in Greece, we can also all patients may participate. And my question is: are there any prerequisites or any restrictions? Let's say how, or more specifically, how can patients and carers help from their side? Are there any steps to be done to prepare the way to start, let's say, a clinical trial? What should we do as carers and as patients?
Maybe I can give an answer, and then I think I have to apologize because my phone has been ringing many times I'm on hospital service today, so I need to go. But let's put it this way. The best thing that you can do to help with clinical trials is, first of all, to be in a patient registry or database, to make manifest to the research community that you are there and you are willing, and you may be willing to participate. There are a number of ways. There are the patient registries, there is the FARA registries, there are the fact of being included in the natural history studies, the BEFACS or the CCRN study, which is called FACOMS. There are specific studies for children as well. So there is a Friedreich Ataxia app that is being developed, and that you can just download on your phone and put your information in it, and that may help you to be in touch with what's going on in the research world and even volunteer for clinical studies. So I think the main thing is to be in touch with the community and to be in some kind of registry, and don't forget the app. I think it's a good opportunity now here to make some advertisement for the FA app. It's going to be a very important tool, and it's available for Apple and it's available for Google, Android-based systems.
Thank you.
Yeah, and the app is also translated into multiple languages, and just to add to what Massimo suggested, I would also say the other thing about being ready to participate in clinical trials is what you're doing today in terms of being educated and kind of really understanding what these treatments are and how they're being developed, and what some of the risks and benefits are going to be, and really thinking about what it might mean to be involved in a clinical trial. And being involved in a clinical trial is a big commitment. It involves going to a research center often multiple times over a short period of time. Sometimes it can be 5 to 8 visits in 6 months. And so it's a big commitment personally for an individual and a family to make to participate. And sometimes it could involve, additional medical procedures. It can involve, obviously, taking an experimental medication. And so really being educated and informed on the therapies that are in development, and also, what's required of a clinical trial is incredibly important, and something that I think all families should take very seriously.
Thank you. Thank you to all.
I needed to go. Thank you all. Efkaristopoli.
He's from the other side of the Atlantic as I need to take the microphone. Okay. But I need to stay here. Okay. So thank you, Jen. Thank you, Professor Pandolfo. Dr. Matt Klein, thank you, and Dr. Myriam Rai, because your commitment to this event is impressive. Thank you for that. It is very important, and this is, I say it as a first step in a close cooperation, which actually means that we can be a reliable and honest partner. And this is something something that we started 4 years ago. We build it daily, and we can improve it. And we are dedicated to invest resources in terms of clinical centers, clinical sites, and of course educating with participating in seminars our beloved doctors in this effort. So thank you all for being here with us today. And I think that this was a great panel. Thank you.
Thank you.
Great.
Before Stephanie.
Stephanie.
Is she online?
She's online, yes.
I'm sorry, break in 15 minutes. It's my pleasure to introduce Dr. Stephanie Shirky, who was supposed to present at the Clinical Research for Innovative Treatments session. She's based in California, San Diego. Stephanie has earned her PhD in Paris, and then she got specialized in stem cells and gene therapy at the Scripps Institute in San Diego. And afterwards, she became associate professor at the University of California, San Diego, at the Department of Pediatrics, the Division of Genetics. And she will speak today about her project on FA development. Thank you, Stephanie.
Thank you very much. Thank you, everybody, and thank you to the organizer for inviting me to this conference. I'm sorry, because of the time change, I had to speak later. But so I will speak about a project that is still in preclinical stage for FRIEDREIC'S ataxia, and it's the use of gene therapy. But in this case, we are calling ex vivo gene therapy because we are using the hematopoietic stem cells. So I just show that not to talk about FRIEDREIC'S ataxia because, everybody knows, but it's just to point out that the issue with FRIEDREIC'S ataxia, as it was very well said by Dr. Massimo, is like you have to fix the issue in many organs in FRIEDREIC'S ataxia. You have to target the brain, the dorsal root ganglia, the spinal cord, the heart, and the muscle. So to do so, the approach we are using in my lab is like using hematopoietic stem cells, which are the stem cells that reside in our bone marrow and that have the capacity of being able to migrate to all the tissues and be able to integrate there for a long time. So we use this approach of autologous hematopoietic stem cell that we use from the patient and we gene modify in a dish. And after the patient goes through a chemotherapy to remove their own stem cells, and then we infuse back the hematopoietic stem cells where they will reside there for the rest of the life of the patient. And this is not science fiction. This is in a clinical trial for many diseases already. And in our own lab, we really went from preclinical to clinical for another disease called cystinosis, which is a multi-organ degenerative disease also. And so, as Dr. Klein said, it's really important to know this, how to go from bench to bedside, and we have the capacity of doing that here. So we went first in the mouse model of FRIEDREIC'S ataxia, and we used the stem cells from a normal mice that were really green, so we can track the cells after transplantation. And we did an irradiation, so it's equivalent to the chemotherapy to remove those stem cells of the mice, and we did an infusion. And what we've seen was very impressive is that the mice that have received, the FRIEDREIC'S ataxia mice that have received the treatment of hematopoietic stem cells had a normal locomotor activity that was equivalent to the wild-type mice that you can see here. I hope you see my cursor. The non-treated mice or the mice receiving cells from the disease mice were not, you see that they have defective activity, defective gait, muscle strength and coordination, whereas the mice treated were equivalent to the wild-type mice. And we checked the dorsal root ganglia where the neurons die, and you see that the two controls of the two disease mice, there is a death of the neurons, whereas the mice that receive the wild-type hematopoietic stem cells, we could preserve the neurons. We also showed that the frataxin expression was increasing the tissues in the brain, in the spinal cord, and that the mitochondrial function was improved. And so that was very, we can tell me, the hematopoietic stem cells will make blood cells. So how can this work? So we know now that the hematopoietic stem cells, the beauty of them is that they don't have barrier. They can go anywhere in the body, especially when there is like damage. And so they can go here in the brain, and you see all the green cells here. It's very impressive, the number of cells that we could see in the brain, in the dorsal root ganglia, and spinal cord. So again, we do one bone marrow transplant and the cells do the job. They go wherever they need to go. And so what we showed is that these cells become macroglial cells, is a sort of macrophages that reside in the brain. And what they do here, they are in red. So the macroglial cells that contain the frataxin that we tagged with a GFP fluorophore can now be seen in the neurons. And this is what we think is that the hematopoietic stem cells become macroglia in the brain, spinal cord, DRG, and they can transfer the frataxin to the neurons to prevent their death. But again, the good thing about these cells is that they can also go to the heart and the muscle. So we don't need to do anything else. They know where they have to go. And if there is damage, they go there. And so you can see again also in the heart all the impressive number of hematopoietic stem cells in the heart and muscles. And again, we show that we have expression of the frataxin, increased expression of frataxin in heart and muscle, and improved of mitochondrial function. So now we want to go to clinic, and we want to then gene correct the cells. However, because the gene, in the case of cystinosis, we add the gene, and it's overexpressed. But we know that in the case of frataxin, the overexpression can be toxic. So we wanted to avoid that. So what we are doing instead is a gene editing approach using CRISPR-Cas9, and we cut the elongation. And so the good thing about that is that most of the patients with Friedreich ataxia have this expansion mutation. So the same approach can be used for all the patients. It's in intron, so we can remove it. It's not affecting the gene. And again, because we just cut the gene itself, then it's expressed in the same manner. So it's a regulated manner. So we showed first in the patient cells that were cell line that if we do that, and this is the optimized version, so if, for example, for this patient 1 or patient 2, you can see that if we do this gene editing approach, we end up with not overexpression, but increased expression of frataxin in these cells from mRNA level, but also from a protein level. So then we went and we use the hematopoietic stem cells from the patient, because now we also want to make sure that there is nothing from the hematopoietic stem cell from the patient that would prevent us to do that. So we used volunteers, who kindly participated to this study. And out of 5 patients, we did this approach of removing the GA repeat. And we obtained a gene editing efficiency going from 12% to up to almost 56%. And in these cells, we showed that the frataxin expression was increased and the mitochondrial function was also improved. So now we went back to the mice, because now the next step is to show in a mouse model that we can have a disease improvement with this approach. And all this step is what the FDA requires. So this is called pharmacology. So we have to show that this works in a mouse model. So the good thing about the mouse model we are using is that if there is no frataxin, murine frataxin, that it has a human frataxin transgene. So they express a frataxin transgene that contains 800 GA repeat. So this is a perfect for our method, because we are using exactly the same reagent than we will use in patient. And so we showed that in their stem cells we can cut the GA expression, and when we transplant these cells back in the mice, we show that we see the gene editing cells that engrafted in the hematopoietic tissues. That's good, but also in the tissues like spinal cord, heart, brain, and skeletal muscle, which are impacted by the disease. But what we were impressed with is that the gene editing efficiency, in this tissue was limited, but when we look at the protein level, the expression of the protein, frataxin, comparing the non-edited mice as control with the mice receiving the stem cells that have been edited, we see a really significant increase of the frataxin protein expression in all the tissue that have been affected by the disease. And we also showed that there is a improved of mitochondrial function, like this is a marker for mitochondria biogenesis, which also is improved in the spinal cord and heart of the mice receiving the stem cells that have been edited, and also the cardiac hypertrophy marker that was increased in the disease mice are improved in the mice receiving the stem cells that have been edited. And so we also, the last thing we've done is that we try to see if we could improve the locomotor activity. So in this new mouse model, the neurological defect is very minimal. So, but we found that the gait shows some parameter of the gait are different in the disease mice, and you can see, for example, the number of steps, because they are smaller, are increased in the mice that are diseased, and they also work with irregularity. And the pressure is also different. But when we look, and again, it's a small amount of mice, so we hope to improve all this, is that we, in the mice receiving the stem cells that have been edited, have an improvement. So we show like disease improvement with these cells that have been edited. So this is, I think, a great step forward to be able to convince the FDA so we move forward for a clinical trial later on. And so this is what we want to do, is take the stem cells from the patient, gene edited them in a dish, put them back, and we hope that they will be able to integrate into the disease tissue and be able to provide the healthy protein in all this tissue. And I just want to finish to thank all the lab members who works in this project, all the patients who participated to this study, all the community and all the funding, the NIH, FARA, who has been a supporter of this project since the beginning, and also FARA Australia. Thank you so much.
Any questions on the results? Did you hear the questions?
I cannot hear, sorry. I don't hear anything.
Hello, I'm George Koliakos from the University of Thessaloniki. And I would like to ask if you or anybody else has tried in a similar experiment mesenchymal stem cells that may have a similar effect with hematopoietin.
Yeah. So that's a very good question. So in the project of cystinosis, we tried in parallel the hematopoietic stem cells, the mesenchymal stem cells, and the whole bone marrow cells. And actually the mesenchymal stem cells have only a short-term effect, beneficial effect. And now we know that these cells actually integrate very short-term into the tissue. They don't stay in contrast to the hematopoietic stem cells. So the mesenchymal stem cells are really good for treating acute disease, but they will not stay for a long time. So it's not good for the chronic genetic disorder like FRIEDREIC'S ataxia.
Thank you.
We do have one more question. It's coming. The microphone's coming.
Thank you. Very nice data. I have a question that, at least to my knowledge, allogeneic stem cell transplantation has not been used for FA's treatment. So I wonder how your approach would provide cure in all affected tissues. I mean, we know that bone marrow stem cells can differentiate into microglia and, of course, restore the brain damage. But I wonder how this can benefit the heart.
That's a very good question. So in the heart and muscles, we have shown, and we have shown that also for cystinosis, and others have shown that, is that they become macrophages, resident macrophages. So it's the same kind, it's the same family of the macroglia cells. And when they reside in the a tissue, and they can transfer the protein to the disease cells, the same way when this happened to in the brain.
Thank you, Stephanie. Thanks a lot for joining today. Thank
you very
to
much.
the previous panelists who are still here. I think we do have a short break before moving to the 30 minutes. Okay, we do have a half an hour break, 30 minutes before moving to the two last sessions. Thank you.